All right. Okay. In this talk, I'd like to go over the anatomy of the ear and the basic physiology of hearing. I'm going to warn you right now, I'm just going to try to do all this in one shot, in one video, so this may get a little lengthy. So be prepared to pause and take a break or get up and get some air or do whatever you have to do. But like I said, I'm going to just try to do this all in one shot. So this may be a little longer than normal. All right. So I want to talk about the anatomy of the ear and then relate the anatomy of the ear into the physiology of hearing. And that's why I print and not write in cursive. So, all right. So essentially, when we talk about the ear, we can break the ear down into three distinct regions. We can break the ear down into the outer ear. Okay, then we can break it down into a middle ear. All right, and then the rest would be the inner ear. Okay. So the outer, middle, and inner ear. All right. Now, a lot of this is, essentially when you take a look at this, if you look at just the basic structure, you can kind of get an idea of how the design of these different parts of the ear can play into the functionality of hearing. All right. So, one thing you have to remember that the ear is essentially, when we talk about the physiologic processes that are taking place, the ear is at the level of the temporal bone, okay, of the cranium. All right. And remember your bony landmarks, okay, the opening of the temporal bone, this is going to be the external acoustic meatus. Okay, some people call this the external auditory meatus. For me, personally, I could care less which one of those two you use as long as it's in the correct context. Okay, so essentially the opening, so the ear opens into the external acoustic meatus of the temporal bone. I just am more familiar with saying acoustic. All right. Now, when we take a look at the outer ear, essentially the outer ear is designed to funnel sound, to funnel sound into the ear itself for the purpose of hearing. Okay, and then you've got, down here, this would be the lobule. Okay, this would be the helix. Okay, and in general, this entire outer fleshy portion of the ear, we would call this the auricle. Okay, the auricle of the ear. All right. Now, again, when you see how this is designed, you can see that this outer fleshy part of the ear is essentially like a funnel designed to move air down and into the external acoustic meatus. Okay, because remember when you're listening to sound, when you're hearing me talk right now or someone talking to you, essentially, but you have to remember this is energy. Okay, except what this is energy of compressed air. All right. Okay, and essentially when compressed air is coming in, you have these, you've got basically waves of pressure. You've got waves of high pressure and you've got waves of low pressure. All right, and I'll talk more about this in a little bit. All right, so this would be high pressure. This would be in the middle would be low pressure. All right, and then essentially, so you've got peaks. So essentially, what you're looking at are peaks and valleys. All right. High and low pressure. So essentially, as this compressed air is floating through the air, the ear will funnel it in, and as this compressed air hits the auricle of the ear, and it will be funneled into the external acoustic meatus. Okay. Now, before I even go any farther with this, let's use this lecture to answer the age-old question: if a tree falls in the forest, will it make a sound? If a tree falls in the forest, will it actually make a sound? Okay, that's something to think about while we're going through this. All right, so now that compressed air is funneled through the external acoustic meatus, and it's going to travel down. You all know what this is. This is the ear canal. Okay, the ear canal. All right, and the ear canal is just a passageway for that compressed air to get into the ear itself. And you know that the ear canal is lined with a secretion called sebum. Okay, or what you guys think of as ear wax. Okay, and the purpose of that earwax is essentially a waterproofing mechanism that is there to keep the ears it does keep the ears moist, but also keep excess humidity from getting in. And also, think of it as kind of being the analog of mucus. Okay, it's a very oily substance, but at the same time, you've got all this. It's very sticky as well. All right, and it's good for preventing parasites and bugs and junk from crawling into your ear and getting stuck in there because it is an opening that's exposed to the outside world. Okay, so it's there to protect the ear as well. All right. And then when you take a look at the middle portion of the ear, you've got what's called the tympanic membrane. Okay, otherwise known as the eardrum. Okay, the tympanic membrane. Okay, very thin fold of tissue that's highly vascular and also highly innervated. Okay, and I'm going to come back to that concept in a second. All right. Now this kind of puts a topic into my head. I hear people say that you shouldn't clean your ears with Q-tips. Okay. And when I hear people say that, it kind of irks me and drives me nuts because why? Why not? Okay, because a couple of pinheads out there stuck Q-tips into their ears and ruptured their eardrums and caused damage and maybe caused deficits in their hearing. They had to go see the doctors, because they're bleeding out of their ear and all that. And I mean, realistically, people, this is just common sense. If you're sticking a Q-tip in your ear, you got to bear in mind, you're not loading a musket. You're cleaning your ear. Don't ramrod the thing in there. And if you feel pressure, stop pushing. Okay, plain and simple. Just put the thing in there, swab it around, get that excess earwax out, and you're good to go. Okay, but I mean, don't candle your ears. Don't dump hot candle wax down there. You don't have to go to the doctor to get your ears cleaned unless you have a specific order or pathology that requires you to do so. I mean, use Q-tips to clean your ears. Just apply, a shred of common sense to this. Okay. So sorry about the tangent there, but so this now essentially we call this the eardrum because when this compressed air strikes the eardrum, I mean, you guys know what happens when you take a stick and you strike the leather upper portion of a drum, it vibrates. Okay, and then as the eardrum vibrates, okay, what's going to happen is it's going to make these three bones called the auditory ossicles move. Okay, and these three, the auditory ossicles, and these three bones would be called the malleus, the incus, and the stapes. Okay, malleus, incus, and stapes, otherwise known as the hammer, the anvil, and the foot plate. And I'll talk about why they're called that in a little while. All right, and then you'll also notice within the middle ear that there is this tube called the eustachian tube. Okay, the eustachian tube is essentially a connection between the nasal pharynx and the middle ear itself, and I'll talk about the importance of that in a second. Okay, so within the middle ear, you've got your eardrum, the auditory ossicles, and the eustachian tube. All right, and then when you get deep into the temporal bone, deep into the inner ear, now you've got these two structures called the vestibular system. All right, the vestibular system and the cochlea. Okay, the cochlea is the organ for hearing. Okay, the vestibular system is essentially for equilibrium. Okay, balance and equilibrium, so it's in constant communication with the cerebellum and other parts of the brain so we can maintain proper balance. And I'm not really going to, in this video, I'm not going to talk much about the vestibular system, but essentially these semicircular canals are filled with fluid, and as we rotate our body and our head, the fluid will shift in these canals, and that will either depolarize or hyperpolarize certain receptors, okay? And as we activate certain receptors based on what direction the fluid is shifting, this nerve, the vestibular nerve, will communicate with certain areas of the brain, and the brain is aware of our body position. So then as a result, like I said, that's how the brain, that's what, for example, when you see a gymnast, they go run and they pull, they vault off that horse, and then they do, 2 somersaults in the air and land square on their feet. That's because the brain, the vestibular system, were in constant communication with each other and are aware of body position. And that's why when people have problems, pathologies or damage or problems with the vestibular system, one of the common things they experience is vertigo. Okay, vertigo is a high-class term for dizziness. Okay, you know, they get nauseated, dizzy, headaches, and all that stuff because now the brain has a harder time understanding body position. All right, and that's about all I'm really going to get into right now in this video with the vestibular system because I want to focus more on hearing. Okay, and then you've got, again, the cochlea. We'll break down the cochlea more in depth. All right, but this almost looks exactly like a snail. All right, and then here you can see, let me erase this a little bit. And then here, associated with the cochlea and the vestibular system, you've got cranial nerve number 8, the vestibulocochlear nerve. Okay, so basically these are two nerves that come together and then travel afferently towards the brain. So again, we can understand body position and also interpret sound. All right, so be acquainted with these structures of the ear. The auricle, the fleshy outer portion of the ear, the external acoustic meatus, the ear canal, and associate all of those with being part of the outer ear. And then within the middle ear, you've got your eustachian tube, your eardrum, and your auditory ossicles. And then within your inner ear, you've got your vestibulocochlear nerve, cranial nerve number 8. And then you've got the cochlea and the vestibular system itself. Okay, and we're going to be breaking down these structures more individually as I go throughout this talk. But again, you need to be aware of all this. And again, make sure you understand that this is all located, again, within the temporal bone. All right, and then here's just some photos I took of an ear model. Again, you can see right here, the nice thing about models is sometimes they're kind of over-exaggerated, so you can see these features. Again, you can see the curvatures of the helix and the auricle itself, and how it's designed, again, to funnel air in through the external acoustic meatus right here. Okay, that compressed air. And then you can see how it will pass down the ear canal and eventually strike the eardrum. All right, and again, it's hard to see in this picture here, and I apologize for that. But again, you can kind of see right here the highly vascular nature of the tympanic membrane, the eardrum itself. And again, you can't really see the innervation very well, but again, this is a really highly innervated and vascular area. Okay, and here you can see the eustachian tube. And let's talk about this for a second, and let's put a little clinical application to this and talk about something called otitis media. Okay, otitis media. All right. Now, the importance of this eustachian tube is essentially to equalize pressure around the eardrum. Okay, around the eardrum. Now, this model you see here would be more representative of an adult ear. Okay, an adult ear. All right, because and you can tell this because the eustachian tube is more vertical. Okay, it's a lot more vertical in young children. As they're in younger children, the eustachian tube is more horizontal. All right, than as an adult. And the clinical importance of that is otitis media is essentially when you think about getting the middle ear infection. Otitis, auto, ear, itis, inflammation, and but should know by now that typically where there's an infection, there's inflammation, and then media meaning middle. Okay. So now, as I mentioned before, that obviously you can't see this on here, but this tube goes down to the nasopharynx. Okay, the nasopharynx, and on the end of this tube there's a thin layer of epithelial tissue that opens and closes in certain situations. And let's think about this for a second. Let's say, I don't know. Let's say you go in a pool. Okay, let's say you go in a pool and you go underwater. And let's say, you like to go to the deep end of the pool. Let's say you go, 12 feet below water. Okay. As you're under 12, and you're just sitting at the bottom of that pool. Okay. There's a lot of pressure from the outside of your head, being forced inward against the tympanic membrane. All right, so the tympanic membrane of the eardrum naturally is going to want to bow inward just due to the excess pressure. So what do you do in this situation? Obviously, the smart aleck would say just go to the top, but let's say you're not. Okay, if you want to equalize the pressure, you guys all know what you do. You plug your nose, you close your mouth, and you try to blow out. Okay, so what you're doing is when you plug your nose and you close your mouth, you're not allowing air to escape as you're trying to exhale. All right, but what you're doing then is you're forcefully trying to blow that air out. You're also forcing You're also forcing the coverings of this eustachian tube and the nasal pharynx to open up, and then you're forcing air up the eustachian tube, and you're building up air pressure on the other side of this tympanic membrane. So then as a result, what you're doing now is you're equalizing air pressure around the tympanic membrane, and you're preventing it from rupturing. Okay, because you know how this feels like when you're in this situation. It almost feels like it hurts, and it almost feels like it's painful within your inner ear, because the eardrum, the tympanic membrane, is sensitive to pressure changes. Okay, again, I mentioned it's a highly innervated area, so as a result, it is quite sensitive to any physical changes that occur within it. Okay, and vice versa. Let's say you go on a plane. Okay, you're just at very high altitude. They always tell you to chew gum. Okay, and again, for the same purpose, except when the higher you go, the thinner the air is, the less dense the air is. So now you're down, you're up high and you're chewing gum. So since there's less air pressure being applied to the eardrum, it's probably gonna bow outward a little more. Okay, so then what you do is you chew gum and you're continuously opening these flaps, and now you're forcing air out again to equalize air pressure around there. Okay. Now, the clinical application to this as well with little kids that have otitis media, gosh darn it. Sorry, folks, the eraser's having a mind of its own right now. So as I mentioned before, in younger children, the eustachian tube is a lot more horizontal. Okay, so now let's say you've got a sinus infection. Okay, you've got an upper respiratory infection, a URI, and now all of a sudden, what's going to happen is you're going to build up a bunch of fluid and mucus and junk, and it's going to be harder for that to drain because it's more vertical versus an adult. I mean, how often do you hear about an adult rupturing their eardrum because they have a head cold? You almost never hear about that, because it's a lot easier for that fluid, just due to gravity, to drain downward that can build up in the eustachian tube. But in these young children, it's a lot harder for it to drain downward, so it sits there and it builds up, and eventually to the point where the eustachian tube can become quite obstructed. All right, and now all of a sudden, if this becomes blocked, you're not able to get air in or out of this tube, and you're just going to build up pressure on one side. Okay, and then those of you who are parents or even yourselves who have experienced this, know what this kid is going through, because it's extremely painful. And then eventually the eardrum, if it's not treated, the eardrum will rupture, and then they'll start to bleed out of their ear. Okay, and then that's why when they treat this, they do what's called a myringotomy. Okay, Myringo is essentially the word root for eardrum, and then otomy is when you make a surgical incision. So what they do is they create a surgical incision in the eardrum, and then they apply a tube in there. So basically this tube is allowing for free flow of air across the eardrum, and then until the infection goes away, and then that eustachian tube opens up, and then they can pull the tube out and then resuture the eardrum, and then all is well. Okay, so that's essentially the importance of the eustachian tube within the middle ear, and then what goes on, with otitis media. All right. Now, also within the middle ear, as I mentioned before, there are three bones: the malleus, the incus, and the stapes. The malleus, the incus, and the stapes. Okay. Okay. Now remember before we said that as sound waves come in, and they strike the eardrum. Okay, so basically we're looking at the eardrum from the middle ear looking out. So what happens is this eardrum is going to vibrate. Okay, the eardrum is going to vibrate. So what's going to happen, we call the malleus the hammer. Oops, forgot how to write the letter M. So that we call the malleus the hammer. We call the incus the hammer, the anvil, sorry. And the stapes, the footplate. Okay, so what happens as this eardrum vibrates, the malleus is essentially, you guys can see why we call it the hammer. It's going to kind of hammer on the incus. Okay, and you guys, remember a blacksmith, you got an anvil. or if you guys like those Looney Tunes cartoons, where they're always dropping anvils on each other. Okay. And then, when a blacksmith is making something, they take this big iron anvil, and then they sit there and hammer on metal on it. Okay. And then what's going to happen is, as the malleus is hammering on the incus, that's going to make it, vibrate and move a little bit. Okay. And then as this vibrates and shifts around and moves, that's going to cause the stapes to shift and move as well. And then attached to this is the oval window of the cochlea. Okay. And I'm going to talk more about the cochlea, but that's the organ of hearing. And the cochlea is a very fluid-filled organ. Okay. So you can get the picture here that as this eardrum vibrates and as these bones move around in the middle ear, that's eventually, eventually the stapes is going to press against the cochlea. And then what do you think is going to happen to the fluid inside the cochlea? If this is constantly, pressing on this and applying force to the cochlea, the fluid's going to shift and move. Okay, the fluid's going to shift and move, and I'll talk about what happens there. All right, and then this right here is what's called the tensor tympani muscle. That's a muscle we use to protect the eardrum. So essentially, when we're exposed to really high frequency sound waves, so basically sound waves that are, so basically that would be high frequency versus low frequency. Okay, so the higher the frequency, the more energy. Because remember, I said that that sound waves essentially are made of positives and negative pressures. Okay, the more positive pressures you have, the more peaks and valleys you have. Okay, so and essentially the more energy that is within that compressed air. Okay, so essentially, so you guys can do the math on this. If we have too high energy of compressed air or sound energy hitting that eardrum, that could potentially cause harm to it. Or it could, but it's not really going to cause a lot of harm to that. It's going to cause more harm to the receptors inside the ear. So then what we do when we're exposed to high frequency energy that's painful to listen to, this muscle contracts and it stiffens up the eardrum and makes it harder for it to vibrate. And it also kind of shifts it medially a little bit as well. So as a result, it makes so as that vibrates less, then it's harder to then it's a lot easier on the receptors within the cochlea, and that'll make more sense when I break the cochlea down more with you. Okay, so that's essentially the middle ear and otitis media and ear infections. Now let's talk more about the cochlea itself. Okay, the cochlea again. So this right here would be the cochlea, and then again this right here would be I'm going to abbreviate this, the vestibular system. Okay. Now, again, as I mentioned before, the cochlea is a fluid-filled organ. That essentially is where you find the receptors for hearing, and the hearing receptors are called stereocilia. Okay, they're these little hair cells. All right, and then there are, when you look at it, this would be like one section or one functional unit of a within the cochlea itself. So then you've got this right here. So essentially there's an outer, middle, and inner membrane within each of these little sections of the ear itself. Okay. And then if we break, and then you notice here, these are what we call spiral ganglion, and then this would be the cochlear nerve. Okay, part of, again, cranial nerve number 8. All right. So now let's kind of break this down a little more in depth. Now, this fluid that's within the cochlea is, depending on what aspect you find it in, is, can be rich in potassium to basically be fairly neutral in its charge and composition. All right. Now, when we take a look at this, essentially what you've got here, you've got what's called the scala. This is the scala vestibuli. All right. Then down here, you've got the scala tympani. All right. And then in the middle here, you've got what's called the cochlear duct. Okay, the cochlear duct. Okay. Now this is important, within this cochlear duct, because this is where the actual organs or the receptors of hearing are located. Okay. These other scala, these are essentially fluid-filled areas. Okay. And these contain, a type of fluid called perilymph. Okay, and then same thing down here, perilymph. All right. So peri meaning outside of. Okay. And then in here, you've got what's called endolymph. Okay, endolymph. All right, so perilymph and endolymph. All right, so this is, again, this is the fluid that we were talking about that's displaced during hearing. Okay, now when we take a look at this, here right here, this, this is, we can break down the organ of hearing. This is called the organ of Corti. Okay, the organ of Corti. All right, and it's broken down into essentially two different membranes. Okay, it's broken down into a basilar membrane. Okay, and which is very, very gelatinous in nature. So it's very, very soft and it's easy to displace. Okay, and then we've got what's called the tectoral membrane. The tectoral membrane. Okay, so essentially, so you can kind of start to figure out what happens here as that stapes starts to slam against the cochlea. This fluid is going to start to displace within here. All right, and then then this gel-like membrane is going to shift, and then on the very, very ends of these, attached to the basilar membrane are where you find the stereocilia. Okay, the stereocilia, the hair cells. Okay, and then you can see at the very ends of the hair cells, at the very ends of the hair cells, excuse me, you can see that there are nerve fibers that are associated with them. Okay, so you can start to do the math here, that as these membranes start to shift around, okay, these hair cells are also going to start to shift. Okay, and as these hair cells shift in certain directions, it's going to cause one of two events, either depolarization, okay, or hyperpolarization. Okay, so essentially these hair cells have very, very oscillating membranes. Okay, very, very oscillating membranes. All right, so basically, if you have enough of that compressed, that compressed air hitting, and you get enough vibrations, then this fluid shifts, and these hair cells shift in one direction. That's going to cause depolarization, and then you'll start to generate action potentials to the to the brain. Okay, if they shift in another direction, that may cause hyperpolarization, and then you'll stop sending hearing information to the brain. All right, so that's essentially, the cochlea and the basilar membrane, and this organ of Corti. Again, I apologize, this is so clustered here. Okay, so again, this is the area we're really going to start to focus on a little while now. Remember, you've got the tectoral membrane that's really stiff, and then you've got the basilar membrane that's a lot more gelatinous and soft. All right, and then embedded within the basilar membrane again are these hair cells. All right, and you've got inner and outer hair cells. Okay, and again, I did a little doodle here. So again, this is where you would find perilymph within the scala vestibuli and the scala tympani. Vestibuli, tympani, and then this would be the cochlear duct. And then here would be the tectoral membrane. And then here would be this blue represents the basilar membrane. Okay, so what's going to happen then is this fluid starts to shift, the basilar membrane is going to go boink, and it's going to bend, and it's going to shift, and then these hair cells are going to move with the basilar membrane. And then as a result, like I said, that's going to cause these hair cells, these stereocilia, to bend in a certain direction. And then when they bend, they're either going to depolarize or hyperpolarize. Okay, if they bend and favor depolarization, again, that's when we generate action potentials, through these into these spiral ganglion. Okay, and then up through the cochlear nerve to the brain. All right, and then, here, again, to retrieve this image from the histology site that I always like, we've got, again, inner and outer hair cells that are located, within here as well. All right. Now, one thing to bear in mind about the endolymph is that I forgot to mention is that it's an environment that's a little different than what we think of as most extracellular environments. It's an environment that's very rich in potassium. Okay, because you think of normally sodium being in the extracellular fluid spaces. It's a little opposite in this situation. Okay, and I'll talk more about that in a little bit. But but I just wanted to bring that up just to get it into your brains while we're thinking about this stuff. All right. So this would normally be the time when I would ask if anybody has any questions, but this isn't a live audience. So let's talk. So let's take everything that I just discussed and put it all together and talk about the physiology of hearing. Okay. So essentially what you've got here, so we've established that sound waves are just compressed air. And you know by now that the sound energy is going to come in and strike the tympanic membrane. Okay, and then the eardrum or the tympanic membrane is going to vibrate. All right, and then as it vibrates, that energy is being transferred, okay, from the hammer, the anvil to the stapes. Okay. Now, the area where the stapes meets up with the cochlea, that's called the oval window. Okay, and you can see that the oval window, its location favors the scala vestibuli. Okay, the scala vestibuli. All right. Essentially kind of one of these outer portions of the cochlea. So, and again, you can do the math here on what's going to happen as the vibrations occur within the tympanic membrane. All right, the auditory ossicles are going to press on the oval window. All right, and then the fluid, the energy from this, the energy from this is going to pass through the fluid, through the perilymph of the scala vestibuli, and then what will happen eventually? That energy will be transferred through the fluid, and then it will shift. Okay, it will eventually come in contact with the tectoral and basilar membrane. Remember the middle portion. This is called the cochlear duct, where you find the organ of corti. Okay, and remember that gel-like basilar membrane. Okay, so that gel-like basilar membrane is going to displace, and then those hair cells are going to bend. All right, and then what's going to happen is as this sound energy passes through the basilar membrane, then the energy will pass through the scala tympani, and then through the scala tympani, and it will eventually hit what's called the round window. Okay, and the round window is again just kind of a thin area of tissue that's gonna constantly bow in and out, in and out, in and out to try to dampen that sound energy so we don't overstimulate the receptors for hearing. Okay. And then you can see, now notice how you see all these nerve fibers projecting from the cochlea out to this spiral ganglion. I'm just going to abbreviate this, spiral ganglion here. Okay, so essentially all along the hair cells that are embedded within the basilar membrane, these, there are nerve fiber projections into, into the spiral ganglion to the cochlear nerve. Now, let me ask you this. Let me erase all this mess here, and then let's erase all this. Let's get all of it. All right. So, essentially, what's going to, now let me ask you this: where do you, when it comes to high versus low frequency sound, okay? So essentially, when we're talking about higher frequency sound, we're talking about sound waves that have a lot more energy, more peaks and valleys, versus very low frequency. Okay, so for example, an example of something low frequency. Well, heck, my voice right now. Okay, my voice. All right, my voice is not I don't have the loudest voice in the world. Let's say compared to a gunshot. Okay, and that's why if you actually shoot guns or target practice, you should always wear earplugs or earmuffs or both, because that's a lot of as you're igniting the powder in the shell, and then that powder is pushing the bullet out of the gun. That takes a lot of energy, and that's gonna basically cause some very big pressure changes in the air around where that gunshot went off. Okay, and if you're very close to that, and you're not wearing any ear protection, okay, you can think about what's going to happen. That very high frequency sound energy is gonna strike your eardrum with a lot of force, and that means it's going to vibrate with a lot of force, okay. And then these ossicles are going to press on the cochlea with a lot of force, and then what do you think is going to happen to those hair cells? They're going to bend a lot. Okay, they're going to bend a lot, and you could potentially damage those if you're not wearing any protection. There's a reason why Ma and Pa said don't put headphones on and blast your music, because it will damage your ears. Okay. So essentially, the more energy, the more sound energy that's coming in, the earlier, the quicker you're going to pick up on that. Okay, so basically, the higher the energy, the more quickly it's going to transfer through the cochlear duct, and the quicker you're going to displace that basilar membrane and the hair cells. Okay, versus versus if you listen to my voice or dull kind of sound, it's going to pass a lot farther through this. Through the scala vestibuli, and it won't displace the basilar membrane until you get towards the apex of it and the very tail end of the cochlea. Okay, and then that dull energy will, it may almost be completely dampened by the time it even hits the round window. All right. And again, you can see, right here, I drew a little visual representation of this. All right, you got this would be your eardrum and your ossicles. All right, so your ossicles vibrate. Okay, they smack against each other, and then this perilymph is displaced. Okay, in the scala vestibuli, and then as this perilymph is displaced, okay, like I said, eventually, let's say a gunshot rang, okay, and you were pretty close to where the shot went off. That fluid is going to be displaced with a lot of force, and it's gonna push against the basilar membrane a lot more quickly. And then that's going to travel, and then therefore that's going to travel towards the round window a lot more quickly. And again, you can do the math here. The higher the energy, the more force that's going to hit the round window with. So that round window is going to bow outward a lot more. Okay. The purpose of this round window is to dampen sound, as I mentioned earlier, because let's say this was just pure bone right here. That sound energy would hit that bone and more than likely go doing and then start bouncing back around through here. And then think of how agonizing that would be if you didn't have anything to dampen that sound energy, and those stereocilia were constantly being bent, and your brain was constantly being told there's sound going on. Okay, that would drive you mad. Okay, versus the low frequency sound energy that's going to pass a lot farther through the scala vestibuli, and then through the basilar membrane, and then back around. Okay. So now I mentioned that those stereocilia, those hair cells, are kind of sitting within a potassium-rich environment. Okay, they're sitting within a potassium-rich environment. So what's going to happen then, and they're connected to one another. So as these hair cells bend and shift, okay, what's going to happen are you're going to open up gates, and then you're going to have the diffusion of potassium into the cell, and then that's going to cause some depolarization of the cell. And then at the lower end of these, so then remember these are, and then remember at the basal aspect of these receptors, there are nerves attached to them. And then basically as you depolarize these stereocilia, then you're going to open up calcium-specific gates, and then you're going to start generating action potentials, down, not down, but towards the brain itself. Okay. And then you can see why, again, if you listen to loud music or you're around loud noises all the time, people experience this, via occupational hazards, if you work at an airport or a factory. I remember going into a factory my dad used to work at a lot. Sometimes when I was a kid, I'd go see him, and there's these, saws that are bigger, they're as wide as the room that I'm in right now, cutting wood. And, those things are going to generate a lot of noise. And everybody in there, including myself, even though I was visiting, had to wear an earmuffs just to protect my ears. Because, if you're shifting that fluid with a lot of force, you're going to damage these hair cells. And then as a result, you may lose your hearing is going to get worse as you age. All right. So that's something to keep in mind. And then the last thing I want to discuss are the projection pathways for hearing to the brain. So essentially what's going to happen as we depolarize, so basically as we start to depolarize those hair cells and we start to generate impulses to the brain, what's going to happen are, so this right here would be essentially be the spiral ganglion. Okay, the spiral ganglion. So, and then this would essentially be the cochlear nerve. I'm just going to abbreviate. I'm just going to cranial nerve number 8. Okay, and those are essentially going to synapse in an area. This is the medulla. That's the medulla oblongata. This would be the midbrain. Okay, and remember this would be the thalamus. Okay, and they're going to synapse with this area of the medulla called the cochlear nuclei. Okay, the cochlear nuclei. All right, and then not all, but some of these fibers are going to cross over to an area called the superior ovulatory nucleus, or ovulatory. I'm sorry, olivary. Okay, where they're then going to ascend upwards through the pons in the midbrain. Now, there's an area of the pons as well. Excuse me. So essentially, as these ascend upwards, some of these fibers may stay on the same side, or they may cross as well. At the colliculi of the the midbrain and so on. Sorry, I said the pons. And then what's going to happen? They're gonna synapse with the medial geniculate, medial nucleus of the thalamus. Okay, and then from there, they're going to project to the auditory cortex. Okay, of the, you guys know what lobe this is, the temporal lobe. So that's where we consciously interpret sound. Okay, so remember we talked about those neuron chains. Okay, first order neuron, second order neuron, third order neuron, and then the fourth would be the processing of that. Okay, now like I said, when it comes to hearing, when it comes to projecting sound up to the brain, not all fibers cross over. So sometimes, so there will be some instances where you will be where the ipsilateral, the cortex will be getting hearing input from the ear in that same side. Okay. Now, one of the other things I wanted to mention as well is, the last thing about this, so you kind of have a basic understanding of this projection pathway. Okay. The last thing I wanted to mention was the processing. How are you able to differentiate between high and low? High and low sound frequencies. Okay. So and it comes back to the processing takes place at the level of the cochlea. Okay. So remember this. So remember before we said that with high frequency sound, that highly compressed air is gonna displace the basilar membrane a lot sooner than later. More towards the base of this than the apex. So essentially, now some of these nerve fibers, kind of stay separate from one another as they meet up with the spiral ganglion and then pass to pass from the cochlear nerve up towards the brain. Okay, so essentially, if you're displacing this basilar membrane a lot earlier, that auditory cortex is going to be getting action potentials, from those nerve fibers, early on in the basilar. Within the basilar membrane versus, the dull aspect of the apex, with dull noise. So that's essentially how we're able to differentiate between, loud noises versus kind of dull, more quiet noises, is these fibers that project from these areas of the basilar membrane towards the brain itself, okay, will stay within their own little individual pathways towards the auditory cortex. Okay, so that's how the brain is able to differentiate between the different pitches and tones of noises. All right, so you're able to tell loud from quiet. All right, so the processing of that takes place at the level of the cochlea. All right, I know that was a lot, but if you folks have any questions on this, you can, feel free to ask me questions, and good luck in your studies, and I will be doing one about the eyes here pretty soon. How long was that?